Hello, welcome to Pharmacology 4343, Pharmacology of the Synapse. My name is Anna Lee. I am an assistant professor of pharmacology, and this week's topic is the nicotinic acetylcholine receptor and the use of genetic knockouts to solve the mystery of what these receptors do at the synapse. In this first of 4 videos, I will introduce you to the nicotinic acetylcholine receptors. The nicotinic acetylcholine receptors belong to the receptor category of the synapse in this organizing schematic. In these videos, we will talk about the genes that encode for these receptors, the molecules that act on the receptors, and how the nicotinic receptors affect the behavior of the whole organism. Nicotinic acetylcholine receptors are pentameric ion channels composed of 5 subunits that surround a central ion pore. One transmembrane domain from each subunit forms the pore. These receptors permit cation flow and can flux sodium, potassium, and calcium. You'll see later in the videos how ion flow can change depending on the subtype of the receptor. Each subunit is composed of 4 transmembrane domains with a large intracellular loop between domains 3 and 4 here. This intracellular loop from the different subunits form the tapered intracellular tail of the receptor. Agonists bind to the large extracellular domain at the interface between the 2 subunits and cause the channel to open due to a conformational change in the receptor that opens the pore. The nicotinic acetylcholine receptors have cis loops, pictured here, which categorize the receptor as a member of the cis-loop ligand-gated ion channels, which also include GABA-A, glycine, and the 5-HT3 receptor, all of which have this motif in common. The brain nicotinic receptors can exist in 3 functional states: closed, open, and desensitized. The closed state occurs when no ligand is bound and the receptor is at rest. The ion channel is not open and there is no current from the receptor. The open state occurs when agonist is bound and the pore is open to cation flow. Brain nicotinic acetylcholine receptors have a very interesting desensitized state. The desensitized state occurs after the receptor has been exposed to agonist for prolonged periods of time. In this state, the ion pore is closed and ligand may or may not be bound. The nicotinic receptors have the ability to be desensitized for very long periods of time, over many minutes. In this slide, here's an example of nicotinic receptor desensitization recorded from transfected cells in vitro. Nicotinic acetylcholine receptors were desensitized by prolonged exposure to the agonist acetylcholine. This drug is then washed out of the preparation, and the receptors are repeatedly tested for their ability to open and conduct ion flow here. This period is 25 minutes long, so desensitization can occur for long periods of time. In general, the higher the concentration of agonist and the longer the exposure, the greater the receptor desensitization. Nicotinic receptor desensitization is an important mechanism in nicotine addiction, which we will talk about in a later week. In previous lectures, you learned about muscle nicotinic receptors in the periphery. The structure of the muscle nicotinic receptors and the brain nicotinic receptors are similar. Both are pentameric cation channels. But there are many differences between these receptors besides their location in the body. Muscle nicotinic receptors are postsynaptic, but brain nicotinic receptors are primarily presynaptic or found on the cell body. The receptors also differ in their subunit composition. Muscle nicotinic receptors are composed of alpha-1, beta-1, delta, or epsilon units, or in the case of embryonic muscle, alpha-1, beta-1, delta, and gamma. In the brain, there are many different subunits of brain nicotinic receptors that have been discovered. There are 8 alpha subunits and 3 beta subunits expressed in the mammalian brain. As each receptor is only composed of 5 subunits, there can be many different combinations that make up a receptor. Finally, the receptors differ in their transmission function. Muscle nicotinic receptors mediate fast transmission that is required for muscle innervation. Because of the presynaptic and the somatic location of the brain nicotinic receptors, they do not directly mediate neurotransmission. Rather, they modulate the activity of other neurotransmitters in the brain. We will touch on this topic in the complex neuromodulation lecture later on in the course.